20 of 20 days of amino acids, and today we're talking about serine. It might look serine, but in the active site of proteases, it can serve as the scissors of these enzymes or reaction speeder uppers that can cut proteins, cut the peptide bond connecting amino acids and break them apart. And so today we're going to talk about how it does it. And so serine proteases, so chemotrypsin, trypsin, is a bunch of them, and they play really important roles in the body and in the lab, and they all use this really cool catalytic triad. And so today, although the focus is on the serine, basically in order to get a tacky, so what it's going to do is it's going to attack the peptide bond, and it's going to like stay kind of stuck, and then half of the peptide is going to get kicked off, then water is going to come and the other half is going to get kicked off. We'll look at it, don't worry. But in order to do that, serine has to get like activated. So in order to attack, so you can think of serine kind of like as the protease scissors blades, but there's like a histidine and an aspartate that are helping kind of pull off the safety blade. So you can think of the proton as kind of being a safety blade. When you deprotonate the serine, then it can then go on the nucleophilic attack the peptide bond and break it. And so what's really cool is that you basically need all of these amino acids. These three key amino acids are like the catalytic ones, the ones that are really, really important for actually making the reaction happen. But then there's other things in the pocket, making like an oxyanion hole that's going to help stabilize the intermediate, make sure it breaks the right way, all sorts of really cool stuff. And so it involves a lot of the different amino acids we've been talking about, and allows us to like connect a bunch of concepts about enzymes, catalysis, amino acids, hydrogen bonding, protonation, deprotonation, all sorts of really cool things going on in a serine protease. And so let's take a look. So overall, what we're going to see, our reaction is going to be something like this. So we have this enzyme, so this reaction speeder upper. More about enzymes later, but they're usually proteins, sometimes a combination of proteins and RNA. In the case of these proteases, it's just a protein. And so you have this big old protein, say this long chain of amino acids that folds up and makes this nice protein. And in part of the protein, we have what we call the active site. And so this is where the things are going to happen. So we have some of the amino acid residues. So basically when amino acids link up, we then call them residues because they're like the residuals that are left over after they combine their amino and acid parts. So now they're no longer like individual amino acids, and so we can refer to these as residues. Although we often just call them amino acids. We can use both of those terms, and I typically use residue. So we have these residues in the active site, and these residues, there's three of them that are going to be directly involved in catalysis, so directly involved in helping speed this reaction up. We're also going to have residues that are important for helping bind the substrate. So a substrate is something that an enzyme acts on. And so in the case of a protease, the substrate is going to be a protein or a peptide. And the protease is going to bind to it and cut it. And so at the heart of these serine proteases is this catalytic triad where we have this serine, a histidine, and an aspartate. So aspartate, you might sometimes see it called aspartic acid, but aspartic acid is when it's in its protonated carboxylic acid form, and we need it to be it's normally in its deprotonated aspartate form, and that's going to be the form that we see it in here. So I'm going to refer to it as aspartate, but you might sometimes see it called aspartic acid. What's going to happen is basically this aspartate is going to help this histidine become more basic. So it'll deprotonate the serine. And this is going to allow the deprotonated serine is then going to attack this peptide, form this covalent intermediate where it's like stuck on half of the peptide, the other half is removed, and then water is going to come in and help kick off the other part. So we'll get more in depth into the mechanism about how this is all going to happen and why we need these three things. We can't just have the serine, we need these three things as well as other things in the enzyme to help make this happen. And so basically what the enzyme is going to do
an enzyme is something that's going to speed up a reaction by lowering the activation barrier. So basically, so if you had a reaction, you have the products have some energy, so the reactants have some energy, the products have some energy, and then in order to go from the reactants to the products, you're going to have to go through a really uncomfy transition state. And basically the enzyme, it can help stabilize this uncomfy transition state and help this reaction proceed. So when we talk, you can think of, if you were talking about like breaking a stick, we can talk about having an enzyme plus a substrate. So the substrate binds, you have this enzyme substrate complex, and then the enzyme breaks the stick and you get enzyme plus product. The enzyme is going to make it easier to get to that breaking point. And it can do this by doing things like helping lower the entropy. So basically entropy is like disorder or randomness. And so these molecules are moving all around and you need them to collide in the perfect way in order to react. And so the enzyme can help hold them together so that they can find each other more easily and then they're in the right position. There's also like the solvation cell. So if we think of those amino acids, if they were out on the surface of the protein, they'd be like surrounded by water, and the water would kind of be hiding them from one another, forming like hydrogen bonds and stuff. But when you're in the pocket of an enzyme, you can have this like solvent exclusion. You can have this water exclusion. Basically, the water can't get in there as much. And so you're going to have the amino acids be more exposed and like attacking towards one another instead of attacking towards water. But we'll also see that water can play important roles. They'll also stabilize awkward transition states, as we'll see. So you don't want to stabilize like just the substrate bound, because then it would have like no motive to change or anything. And you wouldn't want to stabilize just the product bound, or else you would never release it. So it's really by stabilizing that transition state that enzymes are able to help the reaction proceed. Another important thing about enzymes is that there are catalysts, which means that they're not, by definition, they can do this over and over without getting like used up. So at the end of the process, we need to have it in exactly the same state as it was when it started in order to, so that it can do this again and again. And so we'll see that in the case of the serine protease, we're actually going to get like a covalent intermediate. We're actually going to have like part of the peptide is going to be stuck onto the protease. We're also going to have some proteination changes and stuff, but we'll see at the end of the reaction, it's all going to be restored to the state that it was in before. If you look at these amino acids that are involved, we talked about how histidine can act as a base. So a base is something that takes a proton. It can act as a base, become protonated, and then it's in this positively charged state. So histidine, however, is not a very strong base. It has a pKa of about 6, so much more on this in my previous posts. But basically, this means that at a pH of 6, half of it is going to be protonated and half of it is going to be deprotonated. If you go to a higher pH, that means that it's more basic of a solution, so you have less free protons around. Histidine is less likely to be protonated. And so in our bodies, where we have a pH of about 7.4, we're going to have less, we're about 1 above the pKa of histidine. And so we're going to have about, only about 9% of it is going to want to be protonated. And so how is it that we are going to get this histidine to protonate, and not just to protonate, but to get its base, not from solution, but from a serine? So serine is not even on our list of these like sometimes charged amino acids, because serine, it has a pKa. So if you have, so for an acid, that's something that donates a proton. A strong acid is going to have a low pKa. We're basically always going to be at a higher pH than the pKa, and so it's almost always going to be deprotonated. With serine, we're looking at a pKa of about 13. So it's going to be very, 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 very much in the protonated state. And it would normally take like a strong base in order to pull it off. And we just talked about how histidine is not that strong of a base. So how are we going to do this? This is where the aspartate is going to come in. So aspartate, on the other hand, is a stronger acid. So it has a pKa of around like 3.7. So it's going to be predominantly in the deprotonated state. Why is this? So it's a lot easier to deprotonate a glutamic acid or an aspartic acid than serine, because aspartic acid, in glutamic acid, they have this carboxylic acid group. When this deprotonates, you get this carboxylate group. And this form is what we call resonance stabilized. So basically we can draw the double bond to the oxygen to like either of these oxygens. And the true thing is that it's more in the middle, like the charge is shared throughout here. This is called resonance stabilization. And as the name stabilization suggests, it makes this really happy. And so this is very happy in this deprotonated state. It can't get this resonance stabilization in this protonated state. So it likes to be in this deprotonated state where it has this electron delocalization, this resonance stabilization. This makes it happy. And so it's going to have a low pKa. It's going to easily give up that proton. But serine, it just has an alcohol or hydroxyl group. And so this is serine, so it has this hydroxyl group here. It has this methylene linker, this like one carbon with two hydrogens, and then it has this hydroxyl group. And so this is going to be a lot less acidic. And so it's going to be a lot harder to deprotonate this. So it's going to have a pKa of about like 13. So we're almost always we're going to be like way, way, way below the pKa if we're talking about just like the general pKa pH of the body. But the thing is that when we talk about these pKa values, we're talking about just like these free amino acids, just like in solution. And when we talk about the pH, we're talking about the pH of just like the solution. And so pH is like a measure of proton availability. And so when we talk about like the pH of around 7.4 or whatever, we're basically referring to like, well, in general, in your cells, in your blood or whatever, the pH is around that. But when you're talking about, you can talk about like a local pH. So if you imagine you have an enzyme, so these are some structures of some of these enzymes, some of these proteases. And so you have these proteins, and what's going to happen is in your active site, you have basically like a pocket in the protein. And so you have a bunch of this protein is just providing the structure and kind of making this little room in the enzyme where the action can actually happen. And when this is happening, it's like excluding water and other things. And it's like you're putting the histidine like right next to the you're putting that histidine right next to the aspartate, and you're putting the histidine right next to the serine. And so now you're making the conditions where you have kind of like you have basically a different pH than the general solution, and a different like pKa than if it were just out in the general solution. So the pKa is totally like context dependent. And so although we talk about those basic values that we can calculate for like the free amino acids, we have to remember that when we're talking about like enzyme catalysis, we're talking about this protein functioning. We're talking about like a very localized reaction in the greater context of things. And so we can take, so these amino acids can take on functions and stuff that you wouldn't really expect them to. So you wouldn't expect serine to just be like deprotonated if it were just like floating around in a cell, but you can deprotonate serine in an active site of an enzyme. And this is really important because when you do this, what you're doing is you're creating a strong nucleophile. And so a nucleophile is something that basically has more electron density than it can comfortably handle. And so it's seeking out an electrophile, which is something that like wants more electrons. And so often, but not always, a nucleophile is going to be negatively charged or partly negatively charged. An electrophile is going to be positively charged or partly positively charged. Oxygen wants to have 8 valence electrons, so it wants to have this like full outer shell. So it's happy when it has that, and so it has that when it's in that like hydroxylate form or when it's in the hydroxyl form. So it can either be get this octet by, so one of these is going to be shared with hydrogen, with carbon, and then the other one is either going to be just a free electron, so it'll have a lone pair, or it'll be sharing a bond with hydrogen, and you'll get a bond that way. So these are like covalent bonds, much more another. I'm not going to go into it here for the sake of time. But basically, these electrons can share share pairs of electrons to form bonds. And they can do that as a way to fulfill like their desires. So oxygen can bind to like a hydrogen or to a carbon in order to get one of the electrons it needs. If it deprotonates, basically what happens is it's losing a hydrogen without the electron. So the electron from hydrogen is staying behind. So it's still going to have a full octet. So it's going to be happy in that sense. But now it's going to have an imbalance of protons and electrons. So it's going to have this negative charge. And therefore, it's going to be looking for something that's going to it can help share the charge with. And so it's going to be seeking out an electrophile. And we call them nucleophiles because basically the nucleus is where the protons are, so where the positively charged stuff is. And so they're going to be seeking, they love the nucleus of another atom, and the other atom would be the electrophile. And so when we have this hydroxyl group, this hydroxylate, it's going to be very nucleophilic. And so if we deprotonate the serine, we're going to get this strong nucleophile that we can then use to attack. You'll also see similar things happen with threonine and tyrosine and cysteine when they get deprotonated. When a nucleophile attacks something, so a nucleophile can basically act as what we call a base. It can act as a base. So a base is basically a special case of nucleophilicity where what you're attacking is a proton. And so we saw bases before. And so if that's deprotonated serine were to just take a proton back, that would be pretty boring, but that would be acting as a base. And if we say it acts as a nucleophile, so acting as a base is acting as a nucleophile, but it's like a special case of it. If it acts more traditionally as like a nucleophilic reaction or whatever, it can basically add on to something, and that can cause something to break, or it can cause something to stick. There's various things that can happen because so it can like attack something and then something else leaves. And so that's what we are going to see when we are doing this interaction. We're going to get that serine to attack an electrophile. So we have our serine, our deprotonated serine is going to be our nucleophile, and then our carbonyl carbon is going to be an electrophile. And so basically a carbonyl is where you have a carbon double bonded to oxygen. Oxygen is very electronegative. What this means is that when it's sharing a bond, when a carbon is bound to an oxygen, the oxygen is going to be pulling their shared electrons towards it. So the electrons are really in this like electron density you can think of, instead of just thinking about like individual electrons, you have this like electron cloud. And so the oxygen, when you have a carbon bound to an oxygen, the oxygen is going to be hogging more of the shared electrons, so more of the cloud. And this is going to make the oxygen partly negative and the carbon partly positive, and thus electrophilic, and a prime site for attack. And so we see carbonyl carbons be attacked a lot in biochemistry. And here where it's going to be attacking is it's going to be attacking the peptide bond. So it's going to be attacking this peptide bond that is linking up these amino acids. And so this peptide bond is an amide bond. So you have a carbonyl carbon next to a nitrogen. And we're going to see that, so this carbonyl carbon is going to be our electrophile. So how does this all going to work? So basically, you don't want that serine to always be deprotonated, right? Or else it would just like constantly be attacking things because it's very attacky. It doesn't like being deprotonated. It's not happy. It has too much electron density. It wants something to share it with. It wants to attack an electrophile. And so you want to control when that's going to happen. And so basically what happens is that when the substrate binds, the enzyme, so an enzyme, although we can think of it as like having this defined shape, it can like shape change a little. And so when a substrate binds, so a substrate, remember, is the thing that the enzyme is going to act on. When the substrate binds, the enzyme can kind of shift shape a little. And to better accommodate the substrate, as well as to kind of like align its active site residues to attack it. So when the substrate binds, these catalytic residues, so this triad, the residues are going to be brought into position to act. And so this is going to activate it when you want it to be activated. So you only want to activate it if you have the right substrate bound. And so different enzymes are going to have different like substrate specificities based on like what's in their binding pocket. So a few common ones are trypsin, chymotrypsin, and elastase. This good article on like PDB 101, which is a really great resource. And they're all going to cut next to specific residues that they like. And so trypsin, it has this this like red dot here. It has this aspartate in its binding pocket. And so it has this negatively charged residue. So it's going to bind to positively charged, bind to and cut next to positively charged amino acids in peptides. Chymotrypsin, it has this big old binding pocket. So it can bind, it'll bind and cut next to these really big bulky amino acids. Then elastase, it has this tighter pocket, and so it's gonna, it's kind of like hydrophobic pocket, so it's gonna bind next to these like small amino acids. So once you have the right substrate bound, now the enzyme kind of shifts a little, and so all of these residues are going to be in the perfect arrangement to do this. And one of the reasons why you need a perfect arrangement is because this aspartate is going to be hydrogen bonding to this hydrogen of the histidine, and making this histidine more basic and more willing to steal a proton. So a hydrogen bond, we talked more about these before, but this is basically, so this isn't like a covalent bond. This isn't like a bond that's holding together these atoms. These are more like attractions between like a like partial charge attractions between an electronegative atom with a lone pair of electrons and a hydrogen attached to an electronegative atom. So this hydrogen is going to be partly positive. The oxygens are the electronegative atom is going to be partly negative, the lone pair on it. It requires like special geometry in order to be strongest. So in our aspartate, although like the focus is like, well, it's charged, yay. It still has like a lone pair of electrons that aren't involved in the resonance, that can actually be involved in hydrogen bonding. And when we talk about the histidine, it's going to have a hydrogen attached to nitrogen. So nitrogen isn't one of the electronegative atoms, and so it can act as a donor. So this aspartate can kind of like form this hydrogen bond with this hydrogen. And so it's kind of like pulling, you can think of it kind of as like partially pulling away the proton. And so this is going to make this nitrogen like, hey, you're taking my proton, I better take another one. So it's going to make this more basic, so more likely to take a proton. And then it sees the serine right next door. And so although serine is normally not that, is normally not acidic, normally not going to donate that proton, when you have this base right next door that's now been strengthened, you have water excluded from this pocket. So it's not like this can just take a hydrogen from a water that's like right next door, but there's a serine next door, so this can deprotonate this serine. Now you have this really strong nucleophile. Because this oxygen is not happy, this oxygen is really basic, and so it wants to, this oxygen is very nucleophilic, and so it's going to want to attack an electrophile, and so it finds one right in next door. So remember, enzymes are holding everything together right in the right position to interact. They've excluded water, so everything is right in this right orientation, right next to each other. And so what's going to happen is that this serine is going to attack this carbonyl carbon. So this carbon that's having its electrons pulled away by this oxygen. You have this partial charge here. You have this negative charge here. This is going to attack this, push the electrons from this double bond onto the oxygen as a lone pair, making the oxygen negative. So this is called an oxyanion when we have a negative charge on an oxygen. So this is we have this thing called an oxyanion hole. And so basically it's a part on the protein. So another part of the active site of the protein, there's this oxyanion hole that's going to help stabilize this intermediate. And so it's going to stabilize this negative charge that is going to develop on this oxygen, and it's going to help make sure that when when you have this tetrahedral intermediate resolve, it's going to resolve in the way that breaks the bond that you want, basically. Throughout the peptide, you have the ability to have hydrogen bonding because you have like these alternating carbonyls and nitrogen and hydrogen. So you have like these alternating donors and acceptors, and so proteins can form these like hydrogen bonding. And we'll see that with the oxyanion hole, we're having this hydrogen on this nitrogen is going to be able to hydrogen bond with the oxygen in that negatively charged oxygen, and this is going to help stabilize it. And we get to talk about another concept we're talking about is that these hydrogen bonds are actually going to be formed with glycine. And so glycine, this poor little guy, it just has an H as its side chain. It just has a hydrogen. And so normally it doesn't get its hydrogen, it can't really, well, it's not this hydrogen that's playing the role, but basically the smallness is going to allow it to be really flexible. And so it's going to be able to take on those kind of like awkward angles and form a really nice kind of like curve that you can have those two hydrogen bonds forming with the oxygen in order to stabilize that oxyanion intermediate. So because glycine is all small and flexible, it makes it really great for helping serve this function in this oxyanion hole. And so I just wanted to say, yay for glycine. It gets to play an important role in an enzyme here. And so we can see that this is it's not the actual transition state because the transition state is much less stable, but you have this intermediate. So you can think of it kind of like being a quasi-transition state, and this being stabilized by the enzyme, which is what we said that enzymes do. They stabilize that transition state, make it easier to get to that transition state, so you have a lower activation barrier to get there. And so now, though, this is really awkward. So you have this like tetrahedral intermediate where you have a charge, you have all this weird stuff going on. And so this is going to get resolved by breaking one of these bonds. And it's going to break the bond. It's going to break this bond right here. So basically when we broke the carbonyl for the first time, it was because we were doing this oxygen from the serine was attacking here. Now, instead of just re-breaking that, which would be really boring, what we're going to do is we're going to break it this way. So we're going to break this peptide bond. So we're going to break here, and this is going to, the lone pair from the bond is going to come and form a carbonyl carbon here. So now you're going to have your peptide covalently attached. This nitrogen is going to take the proton from this nitrogen is going to take the proton from the histidine. So remember, we have to regenerate everything with an enzyme. And so it's going to take this histidine, the proton that the histidine had taken from serine, the nitrogen is going to now take it. So you get this nitrogen protonated on its way out. And now you have the peptide covalently attached to the enzyme. So the first part gets released, but the N-terminal part is still stuck. And so now we need to resolve that. How we're going to resolve that is with the help of water. So I said we exclude water, but there is some water, we just in a very controlled manner. And so basically now this you still have this. So this histidine, it had its proton stolen by the protein fragment that was leaving, the peptide fragment that was leaving. It stole the proton back. So now this histidine is still basic because it's getting it's still forming that like hydrogen bond to the aspartate. But now it doesn't have that serine right next door, because now that serine's attached. Well, I mean, it has a serine right next door, but it doesn't have a protonated serine that it can take a proton from. And so now it takes a proton from water. When it takes a proton from water, or now it becomes protonated. And what you did was you deprotonated the water. So now this water, this hydroxylate group, is going to act as a nucleophile. So just as we saw how if the oxygen were attached to a carbon, so if you had an alkoxyl group, that would be attacky. It also happens when you have this hydroxide ion, and so this is going to be attacky, nucleophilically attacky, and so it's going to nucleophilically attack. So it's going to attack this carbonyl carbon, just similarly to how we saw before. This poor carbon's being attacked on all sides. But in this case, what happens is you have a different tetrahedral intermediate, and it's going to collapse. It's going to collapse in a way that's going to make this give us this carboxylic acid, and this serine is going to be regenerated by taking that proton. So before we saw that the leaving group was going to take a proton, and here we have that the serine is going to take the proton. Either way, we end up with this carboxylic acid and this serine regenerated. Everything is now regenerated, and you can do this all again. So we can classify proteases based on like where they cut. So an endoprotease is going to cut in the middle, and an exoprotease is going to cut from the ends, as well as how they cut. So some of them do their own cutting in 2 steps with covalent intermediates, where the peptide is going to get permanently stuck to them, like we see with serine proteases. And cysteine proteases and threonine proteases. And so when we're talking about these proteases, what we're talking about is we're not talking about what they're cutting next to, although that is one of the ways that we can classify proteases. But we're talking about what amino acid in the active site is going to attack. So here you're going to have the serine or the cysteine or the threonine attack. Others are going to activate water to do the cutting for them. So you have like a single step without a covalent intermediate, so without it getting partly stuck on. And it's classified by like what gets the water to react by removing a proton, deprotonating the water. So this could be an aspartate or a glutamate or a metal ion. So cysteine and threonine proteases are going to work similarly to the serine proteases. In the case of a cysteine protease, you actually don't need the aspartate because cysteine is more acidic, so it's easier. The histidine itself can do it. And with aspartate proteases, they're actually going to get water to do the cutting for them without having the peptide actually stuck in the process.